And greetings. I think that green screen's not working so well. It's really slowing everything down. I'm going to turn it off. So today I'll be talking about something that I'm going to tie up some loose ends. Some people on the internet, like this dude named Emo, I've since banned him. The guy's just pathologically incapable of understanding, even when I've explained something multiple times. He has falsely accused me of saying, oh, I'm acting like I made some great new discovery that no one else knew about the protein orchard. That's actually not the case at all. I have been pointing out that this is so obvious, and evolutionary biologists are not talking about the problem. And so I find it very disturbing that, let me see what's going on here. I find it very disturbing that something this obvious is not talked about. So Dr. Dan Stern-Cardinal and others have said, no, they don't believe that all major protein families / genes, really the genes that code for the proteins, for strictly speaking, share a universal common ancestor. And I said, well, then how do they evolve? And the standard answer is, well, we don't know. And I recently read an article about eukaryotic evolution, and they're saying, well, these deep evolutionary questions we'll never know. We'll never have clear proof of what happened. The implication being, but you still got to believe it. I just laugh. I said, how is this science? That's just a faith statement. And I've argued multiple times this flies in the face of probability. And I'm actually going to show, using some databases, how some of these probabilities can even be somewhat estimated, how difficult it is to converge on it. And this kind of came about because I'm going to be trying to teach a intelligent design course, trying to develop it, and teach it. I hope Professor Carissa Shipman, you saw her, she was my debate partner maybe about a month ago, and we were just bumping around the idea. This is something that's been on my heart for about now almost 20 years. And there was a poll that I conducted it was informal, but it was released in the prestigious scientific journal Nature, April 28, 2005. It was embedded inside the article that featured me on the cover story. And in that poll, we discovered 75% of the students at James Madison University would be interested in taking an elective in intelligent design, creationism, and/or both. That's a huge number. That's a huge number. And even if it were off by a factor of 10 and only 7.5% wanted, in a campus of 10,000 people, that's still a big number, okay? It's still 750 students. That'd be enough to pack out a lot of classes. So the interest is there. Sadly, this is not taught well in seminaries or Christian schools. They say they cover intelligent design, and just like, that it is so anemic what they teach in seminaries regarding intelligent design as to almost be worthless, in my opinion. To really be able to engage the culture, you have to argue it at a level where you can take down a university professor in a debate. And it's just not going to cut it. If your audience is the university, you have to argue at the university level. You can't be arguing, for some from some congregation that is unsophisticated and wouldn't really care, and they'll just hear a 5-minute explanation, and then I mean, I know a pastor, he just thinks, oh, just a 3-minute explanation ought to suffice to refute evolutionary theory. I'm just like, dude, I give up. I mean, this is why people are leaving the church. You're not giving anything of substance for an answer when students are hungering. So let me, oh man, there's a lot of construction going on late at night. Yeah, it's almost midnight. They're repaving the roads because there's no traffic. So this is kind of an ideal time as far as traffic wise, but it is so noisy outside. You might be hearing the purr of engines and stuff. So let's get to it with the protein orchard. For the longest time, they said, oh, evolution antibiotic resistance is proof positive we can evolve, microbes into human beings. You just accumulate all the mutations. The problem with that is not that apparent until you really study the molecular details. What creationists typically have said over the years is like, well, it's still, that E. coli evolved antibiotic resistance, but it's still an E. coli. That was kind of in the right direction, but it still felt really lame. But now that we have cheap genome sequencing and researchers have improved their techniques to be able to do this quickly and efficiently, we actually do know the molecular details. So you've seen some of my arguments for the protein orchard. It's just blatantly obvious that certain family of proteins cannot evolve from a common ancestor. Even Dr. Dan had to concede that after I showed him that. He may have already known it, but even computationally, you could see these islands. These proteins exist kind of like islands in a sea of non-functionality. You'll have all these possible sequences, and all the sequences that make a functional protein in biology are all clustered together in families. And then outside of the family, there's just like not the sea of non-functionality until you hit another island or oasis or whatever you want to call it that has a functional protein. So my favorite example is the collagen versus the zinc finger. You could clearly see that one cannot evolve a collagen from a common ancestor of some zinc finger. It just doesn't make sense. The evolutionary biologists will agree with me. So why was that an important concession? The saying goes is, look at antibiotic resistance. That's just able to evolve all sorts of things, and all it has to do is accumulate over time all these changes, and out comes a creature. I have discovered a paper that is showing it. The evolutionists at best are far from proving their point because of the molecular details, and at worst they're just dead wrong, and I'd argue they're dead wrong. I don't know if I already mentioned Emo. He's such a dodo. He never gets that. I was never saying that I suddenly discovered something that no one else already knew, that there's a protein orchard. Everyone knows this. The problem is evolutionary biologists have not come to terms that this is devastating to be able to evolve major groups of creatures. I mean, you can't have metazoans. Metazoans, I think, are animals that have embryos. You can't evolve metazoans unless you have collagen, and collagen is a complex protein. You have to have a very punctuated gargantuan change in so many things, including collagen, to make a metazoan. Because first of all, a collagen to be functional has to have a lot of integrated parts in the whole system. And we're dealing with a multicellular creature that has to suddenly have collagen in it, and that's to coordinate all sorts of things in the extracellular matrix. The extracellular matrix has to be able to communicate through the membrane of the cell and then through the nuclear pore complex all the way into the regulatory systems inside the nucleus and then back and forth. This is nuts to think this can evolve in gradual steps from a system that didn't have a collagen apparatus. So that being said, I found this paper, and I'm gonna let me share my screen. I had to do some searching. Let me put it up here. One moment. Type 2 topoisomerase. Mutations in Fluoroquinolone-Resistant clinical strains of Pseudomonas aeruginosa, isolated in 1998 and 1999. Role of target enzyme in mechanism of fluoroquinolone resistance. Now that was a mouthful. Oh my goodness, that was a mouthful. But first, it's a type 2 topoisomerase, also known as Gyrase A, Gyrase B, and PAR-C. And I know I'm gonna get technical real fast. I mean, if I were teaching the class, I'd have to kind of have a feel for where the students are and how much background material. But since this is the first time I'm just putting out on the net, I'm assuming the audience is really sophisticated. This is a nerd torture session, obviously. So one way to begin the search, just to understand this, and I'm just going to show how I do this. So we have the names of some of the proteins like Gyrase. Yes, I know, it's just, they named the protein with just 4 letters, Gyrase, which stands probably for Gyrase A. And the creature is Pseudomonas aeruginosa. So let me just show how it's done. So what I do is I go to uniprot.org, and it says find your protein. I'm going to say Gyrase Pseudomonas. There it is, Pseudomonas aeruginosa, and it's called DNA gyrase subunit A. And so I'm just going to click here, and you'll see why I'm going into this. We want to look at the spelling of this protein. And there you have it. There it is. That's the alphabetical spelling of it. Thankfully, they lay out the coordinates. So, I mean, for example, it puts a 10 here. That means that P, the proline, if you could see my cursor, that P is at position 10. And then this S, the serine, is at position 20, etc. And obviously then this Y, which is tyrosine, will be at position 21. You can sort of figure out how to, it's nice, you could figure out exactly what letter is in what position in the primary structure of the protein. And so this gyrase is 920, roughly 920 amino acids long, 21, 22, 23. Let's see if it says it somewhere here. 923 amino acids. So where I want to go with this is the evolutionists will crow and said, look, we evolved a superbug. It's resistant to this antibiotic. It turns out that it only changed 2 amino acids, and it developed this antibiotic resistance. But we know that because topoisomerase/gyrase is so critical to function, it still has to be a gyrase, or it still has to be a topoisomerase/gyrase for the creature to be alive. You disable the entire enzyme unless there's a backup copy somewhere else. The creature's dead. So it's not going to evolve stepwise to be another protein. So it's at best, it's unproven until the Darwinists actually study the molecular mechanisms before they start proclaiming victory. They better be able to understand the mechanisms that induce antibiotic resistance. Because if that involves taking the protein and making one that is what we call a homolog, and I'll define homolog a little later. Homolog is really basically the same thing with a slight variation. So maybe in the man-made world, look, you have a blender, you have a piano, you have a car. You can make modifications to a car and it's still a car, but it's never going to be a blender or a piano. And you can't make gradual modifications from going to a piano to a car. Now that may seem extreme, but let's take even a simpler example. You're not going to evolve a bolt into a nut. You might be able to make changes to a nut, even conceptually, and it will still be a nut, but it's not going to be a bolt. There are probably some pathological ways you could work around that, but that's the spirit of the argument. You can't evolve a radiator into a piston. It just doesn't make sense. Geometrically, it just doesn't make sense. You're not going to evolve a piston into a gas tank. You're not going to evolve a tire into a battery. There might be a group of designs that are still tires, but you can do slight modifications, but it still has to be, it's going to be either a tire or something else. There's not an intermediate between a tire and a car battery. In a mechanical sense. At the molecular level, you definitely see that with proteins. A protein's going to be a topoisomerase. It's not going to be a helicase or a polymerase subunit or a collagen or a zinc finger protein. Just like in the man-made world where we have parts of a car that are very distinct and will not undergo gradual geometric changes to be something else. You have these islands of isolation, same thing in proteins. And that shouldn't be surprising because proteins are mechanical parts and have a lot of mechanical properties. They have a defined geometry that makes them fit with other parts. The more you study proteins, you'll see this. So if you evolved antibiotic resistance by making a topoisomerase/gyrase into a slightly different topoisomerase gyrase, that's not adequate to make a quote-unquote change on the order of what we would, what we have colloquially thought as macroevolution, whatever that stupid term means. It would have, I have to come up with a better term, but it had to be something that is at the molecular level just astonishingly and hugely different than what it was before. So I showed the spelling of this topoisomerase gyrase, and let's look at the paper. And this was a tough experiment to carry out. let's look at the paper. It's a great paper. There's another paper that I linked in the comment. In the video description. It's the one that led to this paper, me finding it. Because it talks about something called this QRDR. It's quinolone resistance determining region. And so what is that region? You need to go into the paper, and it identifies the coordinate of where that amino acid is. And if it is changed, like say the threonine in position 83 is changed to isoleucine, alanine, or the aspartic acid is changed into a tyrosine, something like that, it will evolve antibiotic resistance. Now, these are only 2 amino acid changes out of a sea of 923 amino acids. So just bear in mind, this is position 83 and position 87 in Pseudomonas aeruginosa. And I'm going to show then what I did is I typed in Gyrase A for Pseudomonas aeruginosa, and you could see the letters spelled out here with coordinates. Now I'm going to zoom in into this region here, into this region. Yeah, into this region. I did this before the show started. And sorry, there it is. I zoomed in. And so in position 83, which I highlighted in red, and also position 89, highlighted in red also. So a T stands for a threonine and a D for aspartic acid. And again, that's what we're looking for. 83 and 87. I'm sorry, it's 87, not 89. So position 83, position 87. So 83 should be a T, 87 should be a D. Yeah, I know. You would think that aspartic acid would start with an A, but unfortunately another amino acid called alanine uses an A. So they just assigned arbitrarily, I guess, D to aspartic acid. And then I showed, there it is, the 3 and 9, position 83. That's position 84, 85, 86, 87, and that's aspartic acid. You mutate these, and it develops antibiotic resistance. Now, as I said, it's still a topoisomerase / gyrase. And how can we know that? There is this thing called the conserved domain database. So let's go back to this entry in Uniprot. That's disturbing. I was in such good shape. So if you go to this entry in Uniprot, it will tell you where you could find a little more information about that protein in Genbank. So I'm just going to click on this. It has an identifier AAA68625.1, and I'm going to open a link in a new tab. And there it is. And it says identify conserved domains. So this is again the gyrase unit, gyrase A subunit. It's a topoisomerase. I'm going to identify conserved domains. I'll show you what that means. So this gyrase in, let's see if I can make this bigger. This gyrase inside Pseudomonas, the typical one, is listed in the first row. in the second row is kind of a standard template of what we would expect a gyrase to look like. It will have a lot of these common elements. All the gyrases across species will follow a general pattern that's similar to this, although not exactly. And it's going to compare it amino acid by amino acid till you, for all of these amino acids inside each of these, inside the protein. It's going to compare it against the baseline template. Now, what's the probability that it would be that similar? So there's the improbability, you can calculate the improbability that it's going to be, what's the improbability that random chance would generate something that will resemble the basic elements of a gyrase / topoisomerase. If you raise 2 to the 1556.16 power, that's a big number. So that means the odds of random chance making it that similar to a gyrase / topoisomerase is 1 and 2 to the 1556 power. What would that be? Like 10 to the 500th power? It's astronomically remote. And even if I were to flip, as it was in the case of antibiotic resistance, where I flip, let's see, that's 80, number 84. Yeah, if I flip the threonine and then I flip the, I think, is that a D? Yeah, the aspartic acid here, then it develops antibiotic resistance. But, the evolutionists will say, well, it'll gradually accumulate over time. The problem is there has to be limits to how much change this thing can tolerate before it stops being a gyrase. And when it stops being a gyrase, because this is a life-critical function, when it stops being a gyrase, the creature dies. So one can't invoke the examples of developing antibiotic resistance as some sort of analogy to how you're going to develop non-homologous proteins in stepwise fashion. You can, hypothetically you could, and you do in the case of antibiotic resistance, evolve one protein into a homolog into one of its homologs. But that doesn't solve the problem of making non-homologous proteins, or what we would call orphan or taxonomically restricted proteins. Things exactly what Dr. Dan and I were talking about, the sort of proteins that don't share a common ancestor with any other protein. So in this case, clearly you have a common ancestor. You have the ancestral gyrase and then its descendants. That's very clear. What we're talking about is how do you evolve proteins that don't have a common ancestor to anything else. You can't use this example of evolving antibiotic resistance to be good explanation for how such things originate. One would have to suppose that, one example would be something in the non-coding region starts to change. Somehow it gets selected for, which is outrageous if it's not getting coded for anything. It would be selected for its regulatory properties, which isn't the same as what it would be if it were a coding segment. This is just nuts. You guys who understand biochemistry know what I'm talking about. And I'm sorry this is deep in the weeds, but this is the level. I'll see these shallow debates where it goes back and forth. And and the guys just have 1 minute to explain. Well, when you get into detail this level, it takes longer to explain it, and the terms become insanely technical. But I'm going through this to show you that when you actually study the details, the case for gradual evolution through accumulation of mutations being filtered out by so-called Darwinian processes, what they call falsely labeled as natural selection. It hasn't been proven to work, and it, at least in the case I'm citing today, it won't work. It won't work. Because, again, all the proteins are like, they're kind of clustered, all the families of the proteins cluster in families, into families. These families are isolated in sequence space, in amino acid sequence space of things that are biologically not functional. I mean, yeah, you could imagine making something functional, but as far as what exists, all those things, all those random sequences outside of these little islands of functionality are just, they don't function. It's very hard to get it to fold properly, and then, much less will it fit and integrate into a larger system, because it's just like the parts of a car. You don't just gin up one part of a car and try to force it into the car. It has shape, and proteins also have shape. To get them to be a part of an integrated system, all the pieces have to fit. They have to be geometrically shaped, and that's, that seems to be lost on a lot of evolutionary biologists. You can't just do these shallow, this shallow analysis of phylogenetic reconstructions and say, well, all the other parts just popped up into existence because we have this one common protein in this complex that is part of every such complex. I mean, it's just like, like, for example, the spliceosome, they'll just see one little segment that's similar across species and say, well, that's proof it evolved. Like, well, you didn't solve the improbabilities. Like a complex like the spliceosome that has 85 to 200 pieces. Yeah, you might find one common piece, maybe just one or two, common to all the spliceosomes, but that doesn't prove how you account for the other 98, 99% that have no homologs anywhere else. So there you go. That was one of my major points. Let me see. Let me wrap up, see if I have any other things. I just wanted to show you can be thorough about this. How long has this taken so far? Like 36 minutes. And I was using a lot of technical terms. So we visited Genbank. Okay. And we talked about conserved domains. We went to Uniprot. We cited a paper. There was one paper that I skipped showing, but it is in the video description. It was the one that led me to this particular paper, but I mean, if you're all curious, you can look it up. It's in the video description. But all this to say that I'm pretty confident at this point that one cannot just blindly invoke, well, since natural selection, or falsely labeled natural selection, evolves antibiotic resistance, therefore you can evolve new protein families into the protein orchard. That, I think I've made the case that that is a non sequitur, and evolutionary biology is built on non sequiturs, on circular reasoning, and equivocation. Oh my goodness. So I've just shown, at least in this case, that antibiotic resistance is not a good explanation for how something like a topoisomerase could evolve into something else. And by way of extrapolation, one protein is just not going to evolve to any other, uh, unless it's a homolog. But that's exactly not the problem you're trying to solve. You're trying to find out ways that non-homologous proteins suddenly emerge. And I don't think they get it, that it's a problem. To them, they just shrug it off, and they just think it's not going to be a problem. So let me just do a little cleanup here of my I have a lot of browsers open. Oh, well, since we're here, I might as well show that other article. Hang on. And again, I apologize for me being a little disorganized, but here is the article that I first started with. And my jaw dropped that after antibiotic resistance evolved, the gyrase was still a gyrase. And I had a correspondence with my co-author, Joe DeWeese, who's a topoisomerase expert, and he communicated, yeah, that's really interesting, Sal. It shows that these are trivial changes, even though it may compromise enzymatic function of the gyrase, it's able to cause antibiotic resistance, but it's still a gyrase. And if they wanted to be thorough, they would actually test whether those changes have an adverse effect in environments that don't have antibiotics or some other environment. It wouldn't surprise me that in order to specialize for one environment, one becomes basically not as well adapted to another environment that it had inhabited before. It is not as well suited for that environment. So again, just because one becomes more specialized for one environment doesn't mean it's going to involve complexity, large-scale novel complexity, and new protein families into the protein orchard. So I think that's everything. So with that, I'm just going to close out. It's been fun. I know this wasn't the smoothest presentation, but I felt I had to at least step forward and put it out there. And in the process, I'll be able to make a smoother, cleaner presentation. So this was just kind of a dry run. And this came about because I'm hearing a lot of people just keep citing antibiotic resistance, and they think that's going to solve the problem of the origin of major protein families in the protein orchard. I'm already, this is the first time I actually studied the problem that of whether the evolution of antibiotic resistance can be an adequate explanation. At the very least, it is called into serious question, and definitely it will fail for select protein families. Whether we can generalize that is another story, but at the very least, the evolutionary biologists, yet again, have shown, I've shown, and the data has shown, that they're premature in their assumptions. And at this point, it is a faith assumption without actually direct evidence. So when they've been saying, look, it developed antibiotic resistance, that is direct evidence you can evolve new proteins in the protein orchard. That's false. That's false. It's never been proven. It's an assumption. Just because something changed doesn't mean that you're creating new taxonomically restricted non-homologous proteins. And I'm actually rather upset and disgusted that they are making those leaps, that they had just blindly assumed that new radical non-homologous taxonomically restricted proteins just popped into existence. And we probably really didn't know. There are probably people that really believed that. But that has to stop because it's not been proven. And that's also a testable claim. When it's not going to make complex proteins, there might be novel peptides that are short length, like say 10, 20, but not 1,000. And not only that, if it's a sufficiently complex functional protein, it won't evolve in gradual steps. It can't, as a matter of principle. And I've discussed that in other videos. So I think looks like we're done. I want to thank everyone for joining me. It was a lot of fun. I am totally worn out. Oh my goodness, I'm so tired. Don't know why. I'm just so tired. Anyway, God bless you all. May God's blessing fall upon all those who love our Lord and Savior, Jesus Christ. Take care, all, and God bless you.